Hi there! Today we're going to talk about humoral immune response. Have you ever wondered what is immune and how our immune fight with pathogen in our body? Immune refers to being protected against infection, disease, or harmful substances because the body's immune system can recognize and defend against them. The humoral immune response, which is a critical part of our body's defense system, its main function is to eliminate extracellular pathogens, things like bacteria and toxins that are circulating outside of our cells. This response is primarily mediated by B lymphocytes. These B cells develop in the bone marrow where they mature and prepare for action. B lymphocytes are triggered by antigen to proliferate and differentiate into plasma cells and memory B cells. The highly productive plasma cells produce antibodies. It's important to know that this whole process starts when antigen binds to the B lymphocyte receptor. As we just mentioned, once a B cell is activated, it undergoes differentiation and splits into two distinct, highly specialized cell types, which is plasma cell and memory B cell. Plasma cell. These cells are essentially antibody factories. They are the effector cells that are actively producing and releasing vast quantities of specific antibodies. Next, memory B cell. These are the cells responsible for immunological memory. They are long-lived, dormant cells that remain in circulation throughout the body. Their purpose is simple but vital, to ensure that if the body encounters the same pathogen again, the immune response will be much faster and much stronger, offering long-term protection. To get the humoral immune response started, the body first needs to properly process the threat, which are specialized to engulf and degrade the pathogen into antigenic fragments. Once the pathogen is broken down, the antigenic determinants are presented on the cell membrane of the dendritic cell. This presentation happens through a specific structure called the MHC class II protein. The next phase involves activating the immune system's main coordinator, the helper T cell. This begins when the helper T cell recognizes the displayed antigen on the dendritic cell. The T cell specifically has a receptor that allows it to bind to the MHC class II antigenic peptide complex. Once this successful interaction takes place, it promotes the secretion of a key cytokine, interleukin-1, by the dendritic cell to activate helper T cell. Activated helper T cell secretes a cytokine called interleukin-2. This chemical signal is a powerful growth factor. It acts in an autocrine manner, meaning it stimulates the same cell that released it and causes the helper T cell to rapidly proliferate and differentiate into two groups, which is long-lived memory helper T cells and more activated helper T cells. A B cell already exhibits a specific surface receptor, typically IgM, which is used for antigen encounters to bind its specific antigen. The B cell engulfs the antigen receptor complex by a process called phagocytosis. Once inside, it degrades the antigen into smaller peptide fragments for internal processing. We now reach the climax of the activation process. The resulting fragments are now bound with the MHC class II protein and are displayed on the B cell's outer surface. The activated helper T cell, which we prepared in the previous steps now, binds to this displayed antigen on the B cell. This decisive interaction causes the helper T cell to release its key cytokine, interleukin-2, to activate the B cell. The activated B cell undergoes rapid division and proliferation, a process known as clonal expansion, creating a large number of genetically identical cells that all target the same antigen. These identical cells then immediately differentiate into two main types: the antibody-secreting plasma cell and the long-lived memory B cell. The plasma cells secrete antibodies which are released into the system, and memory B cell remain dormant for next infection. The secreted antibodies bind to the pathogen and either block its function or mark it for ingestion by phagocytic cells. This mass action clears the extracellular space of the invading microorganism, successfully eliminating the infection. Finally, the response resolves. Once the pathogen is cleared, most short-lived plasma cells die off. However, the long-lived memory B cell and T cell remain dormant, ensuring the body is prepared for a swift and strong response upon next exposure. That's all from us. Thank you for watching, and we wish you a better understanding of this fascinating system.